Hello everyone and welcome to week 2 of Applied Immunology. This is lecture 2A, the first lecture for this week's module where we'll begin to learn about the innate immune system. In week 1 we focused on higher level conceptual discussions of the immune system and types of immune responses, so this week is our first week where we'll really begin drilling down into these topics at a higher level of detail. Today we'll begin our introduction to innate immunity by introducing primary classes of pathogens and the key goals of innate immunity in addressing infection by those pathogens. We'll then cover physical barriers that exclude pathogenic microorganisms, the different types of innate immune cells, the methods employed by sentinel phagocytes to consume and kill pathogens, and end with the definition of the term inflammation and learn how phagocytes promote inflammatory signaling. As a reminder, this is just the first of 3 lectures this week, so 2B will introduce categories of innate immune receptors and their signaling mechanisms, while 2C will introduce 2 additional layers of innate immunity, which are programmed cell death and complement. As we discussed in week 1, the immune system has evolved in order to defend host organisms from infection by pathogens. In order to learn about the specific mechanisms of the immune system that protect us from infection, we first need to learn a little bit more about the primary classes of pathogens that infect metazoans. As a full disclaimer, this will not be an exhaustive introduction to pathogens. For that, you should probably enroll in a microbiology course. But for immunologists, we primarily categorize microorganisms in terms of the cellular niches that they occupy during their life cycle, as well as the molecules that they produce that can be detected by the immune system. So those 2 metrics will be the primary ways that we compare pathogen categories in this course. First, let's go over these major categories. We can separate the majority of infectious microorganisms into 4 major classes: viruses, bacteria, fungi, and parasites. Here we have bacteria split up into intracellular and extracellular bacteria. First, let's look at viruses. These are the tiniest microorganisms and basically consist of nucleic acids with various types of envelopes or coatings. Since viruses are such minimal units, they don't have organelles for carrying out cellular functions like energy production and replication. These are obligate intracellular pathogens, which means that they absolutely require intracellular infection of host cells in order to carry out their life cycle. We categorize viruses based on the types of nucleic acids that they use to encode their genome. So there are DNA viruses, which, like eukaryotic cells, encode their genome through DNA. And this includes things like pox viruses that we introduced in week 1, as well as herpes viruses, such as herpes simplex virus or varicella, which is the causative agent for chickenpox. There are also RNA viruses, including retroviruses, such as HIV, which causes AIDS, and of course, everyone's current favorite, SARS-CoV-2, which is a coronavirus that's obviously responsible for the current COVID pandemic. RNA viruses can be further subdivided into single-stranded versus double-stranded viruses, which we'll discuss in the next lecture on pattern recognition. Next, we have prokaryotic organisms called bacteria, which are separated based on intracellular versus extracellular phases of their life cycle. Intracellular bacteria consist of those that are facultative, meaning that they have the flexibility to infect cells either extracellularly or intracellularly upon certain conditions, or obligate intracellular bacteria, which again means that they absolutely require entry into a host cell to complete their replication cycle. Facultative intracellular bacteria include Mycobacterium tuberculosis. This is the causative agent of tuberculosis, obviously, which is a respiratory infection with massive public health burden, as well as gastrointestinal pathogens such as Salmonella species and Escherichia coli or E. coli, which I'm sure everyone here has at least heard of. Obligate intracellular bacteria include species of the sexually transmitted pathogen chlamydia, as well as rickettsia, which is transmitted through insect vectors such as mites and ticks, and Coxiella, which is a zoonotic infection transmitted from livestock to humans, which can cause a severe type of infection called Q fever. Next, extracellular bacteria. I should note that all bacteria can be categorized as either gram-negative or gram-positive, both intracellular and extracellular, but we'll just introduce this here under the extracellular column. Gram staining is an established microbiological technique that evaluates the thickness of a peptidic glycan component of the bacterial cell wall. So gram-negative bacteria have a thin layer of peptidic glycan and therefore exclude staining with a type of dye called crystal violet, while gram-positive bacteria have a much thicker layer of peptidic glycan in their cell wall that absorbs crystal violet dye upon staining. So gram-negative bacteria include Pseudomonas aeruginosa, which causes opportunistic infections in immunocompromised humans, as well as Vibrio cholera, which colonizes saltwater organisms and can cause severe gastrointestinal symptoms upon consumption of contaminated water sources. Some gram-positive bacteria include Streptococcus pyogenes, which can cause skin infections, as well as Bacillus anthracis, which produces anthrax toxin, which I'm sure everyone here has heard of. The third class of pathogen are fungi, which are eukaryotic organisms that can grow intracellularly or extracellularly. Common fungal pathogens of humans include Candida, which can infect numerous tissues throughout the body, including the vaginal tract. This is a form of candidiasis that's colloquially referred to as a yeast infection, as well as the mouth, which is a form of candidiasis that we usually refer to as oral thrush. More severe forms of invasive candidiasis can also be life-threatening. Another important fungal pathogen are species within the genus Aspergillus, which are ubiquitous environmental molds that are a very serious concern when they're found in hospitals, because they can cause life-threatening nosocomial infections or hospital-acquired infections, particularly in immunocompromised patients. Parasites are the fourth and final category of pathogen. So, like fungi, these are eukaryotes, and include a variety of organisms, including protozoans, which are microscopic and unicellular, ectoparasites, these are insects such as ticks and fleas, and helminths. Now, helminths are worms that can be divided further into subcategories, including nematodes or roundworms, and trematodes, which are a type of flatworm. Examples of nematodes include trichinella, which live in skeletal muscle and can infect humans following consumption of infected meat, such as pork that hasn't been cooked properly. This is an infection in humans that we call trichinosis. There's also Nippostrongylus, which is a type of hookworm that infects rodents and is used as an experimental study model. Trematodes include schistosoma, which are commonly referred to as blood flukes, and these are consumed through contaminated water sources and then can attach to the urinary tract or intestines, and they cause a serious disease called schistosomiasis that infects large amounts of people living in places like Asia, Africa, and South America. Now, although we can categorize the majority of pathogens into these four categories, they encompass a really enormous diversity of infectious organisms in terms of the molecules that they express, the stages of their life cycle, their requirements for infection and replication, as well as the types of clinical symptoms that they induce in infected hosts. So given this massive level of microbial diversity, we come to the central question of how does the immune system sort of defend the host from all of these infectious agents? So to begin to answer this major overarching question that we'll touch on again and again in this course, we'll first learn about the processes that contribute to immediate or early immune defense. Well, the first of these are anatomic barriers, which are typically epithelial surfaces that physically exclude pathogens through mechanical, chemical, and microbiological mechanisms. These are constitutive barriers that offer immediate forms of protection against infection. The second process is innate immune activation, which we were first introduced to last week in week 1. These are often induced responses that are engaged rapidly in the first hours and days following initial infection. Now, we've already established that the detection, consumption, and killing of pathogens are key goals of an immune response, which the innate immune system accomplishes through pattern recognition receptors and their downstream signaling, the generation of cytotoxic effector molecules, the recruitment of other immune cells through a phenomenon called inflammation, and activation of the complement pathway. And these are all things that we're going to cover in upcoming lectures. In addition to these four components of innate immune function, we've also learned that innate immunity plays an essential role in stimulating later stage adaptive immune responses through the process of antigen presentation. And this is what enables finely tuned pathogen-specific responses that are engaged several days after initial infection. So if you remember from our introduction to innate immunity last week, the components of these last 2 points represent the primary functions of innate immunity. We'll learn more about each of these processes during weeks 2 and 3 of this course, while for the remainder of this lecture, we'll specifically focus on outlining the anatomic barriers and the contributions of phagocytes to innate immunity. First, let's look at physical barriers to infection. Epithelial surfaces in the body are obviously lined with epithelial cells. Primary epithelial surfaces include skin, gut, lungs, and the eyes, nose, and oral cavities, all of which are color-coded in this chart on the right to match the organ systems depicted here on the left. I'll also add that while it's not included in this particular figure from the textbook, the lining of the reproductive tract in males and females is also an important epithelial surface. Taken together, each of these epithelial sites represent areas where microorganisms from the outside world first make contact with a potential host organism that they're trying to infect, which is why it's so important to have these barriers function properly. The first contribution of epithelium to defense is mechanical, as the tight junctions joining individual epithelial cells prevent pathogens from just passing through this first layer of cells. Additional mechanical components include fluid flow over epithelial surfaces, including air, as well as fluids such as mucus, which can be facilitated by the beating of cilia in certain organs, such as the lungs and nasal cavity. With the exception of the skin, each of these other epithelial surfaces also produces mucus. And so we commonly refer to these as mucosal sites or the mucosa. Mucus can function by trapping debris as well as delaying microorganisms in their access to underlying epithelial cells. And this often allows for their removal as mucus is moved through a given organ system mechanically through processes such as sneezing or coughing. In addition to these somewhat obvious mechanical barriers, the epithelium typically employs a variety of chemical defenses to infection as well. These include antimicrobial peptides such as defensins, Reg3, and cathelicidin that are outlined here, acidic microenvironments such as those that are found in the gut that makes it inhospitable to organisms that aren't adapted to acidic growth conditions, and enzymes that can digest components of pathogen walls. Lastly, all epithelial surfaces in the body are also colonized with commensal microbiota, so this includes things like commensal bacteria, as well as viruses and fungi, that under homeostatic conditions in healthy individuals do not actively infect their host cells, they just sort of cohabit there. And these provide a microbiological layer of defense. This is a competition mechanism where commensal microbes can just outcompete pathogens for access to critical nutrients as well as space to grow on at an epithelial surface. And this provides a really essential additional barrier to pathogen outgrowth. Now, given these physical barriers to infection, how do pathogens actually successfully infect a barrier tissue? There are two main ways that this can happen. One is through physical injury that compromises barrier integrity, such as a puncture wound or a cut or a burn. Now, any of these types of insults disrupt all of the physical mechanisms employed by epithelial surfaces to exclude pathogens that we just talked about. And this grants these pathogens access to the underlying tissue where it's easier to establish infection, although immune cell sentinels are also stationed there and ready to respond, which we'll discuss in upcoming slides. Aside from damage to epithelial barriers, pathogens can also successfully establish infection by evolving molecular mechanisms that override physical barriers. And this establishment of successful infection is commonly referred to as the process of pathogenesis. This table on the textbook lists a variety of human pathogens that have evolved such pathogenesis mechanisms in order to successfully colonize the host upon their introduction to mucosal surfaces. And this can be done through the inhalation of droplets into the respiratory tract or consumption of contaminated food or water, which brings pathogens into the gastrointestinal tract. Once these host-adapted pathogens are introduced to a mucosal site, they can employ any of a number of pathogenic mechanisms to transit through the mucus and bypass epithelial cells to successfully infect underlying tissue. We'll discuss some of these mechanisms in our upcoming Week 3 lecture on host-pathogen interactions. The main take-home point that I want you to focus on with this table is that despite the anatomical barriers to infection that we have present at epithelial surfaces, there are many pathogens that still succeed at infecting host tissues. So let's learn more about the first steps of an innate immune response that are induced or engaged once a physical barrier has been breached. To learn about the early induced responses of innate immunity, we first need to learn about the constituent cell types involved and each of their contributions to the immune response. You should remember this differentiation plot from Week 1, where we learned that almost all innate immune cells are myeloid cells that are derived from hematopoietic stem cells in the bone marrow. So let's learn a little bit more about each of these cell types one by one. First, we have macrophages. Now, these are voracious eaters that I want you to associate with phagocytosis, or the uptake and breakdown of extracellular particles. Note that macrophages are found in tissues, but can be formed constantly from circulating monocytes in the blood that enter a given tissue, where they then differentiate into macrophages. Second, we have dendritic cells, or DCs. Now, DCs can also phagocytose extracellular material, so they are phagocytes, but not to the same magnitude that macrophages do. So I want you to associate dendritic cells primarily with the process of antigen presentation, which we'll cover next week. Now, together, both macrophages and dendritic cells are considered professional antigen presenting cells, or APCs. Next, we have mast cells. These contain intracellular organelles called granules, which are filled with inflammatory molecules such as histamine. Mast cells are important during parasitic infections and influence mechanical defenses such as muscle contraction in the airway. And we may cover these briefly in the future as we discuss how their functions relate to the coordination of anti-Helminth responses. Eosinophils and basophils are two types of innate immune cells that play important roles in immune responses to parasites. And interestingly, these can also be inappropriately activated in the context of allergy. And we'll discuss these more in future lectures. Lastly, neutrophils are another key subset of myeloid cells. So like macrophages, they phagocytose extracellular debris. Today we're going to focus our discussion of innate immune cell types on phagocytes. Again, these are primarily macrophages and neutrophils, since these cell subsets are responsible for sampling the extracellular space and digesting phagocytosed material such as pathogens. So you can start to imagine why these are important sentinel cell types that are some of the first to become alerted to the presence of infection. Macrophages and neutrophils make up the primary phagocytes of the immune system, but differ in some key ways. Now, macrophages are seated in peripheral tissues or established in peripheral tissues during embryonic development, and most organs have specialized versions of these tissue-resident macrophages that are specially adapted to that tissue microenvironment. Now, in the brain, these are called microglia, while in the liver, these are called Kupffer cells. The tissue-resident macrophages are long-lived and can be found in pretty much every other organ, including the epithelial barriers that we just discussed. Note that while macrophages are typically viewed as being found primarily in tissues, they are constantly replenished by monocytes that circulate through the blood, where they can exit to enter tissues and then differentiate into macrophages in response to certain differentiation signals. Neutrophils are the most abundant type of immune cell or leukocyte, and in contrast to macrophages, have an extremely short lifespan of only a few days. They're primarily found circulating in blood following their development in the bone marrow, and don't typically enter peripheral tissues under healthy homeostatic conditions, although they are recruited to tissues in response to signs of infection. On a side note, that's kind of a pretty gross type of fun fact for this class. Since neutrophils have such a short lifespan and they're some of the first immediate responses to signs of infection, dead neutrophils are actually a major component of pus, which is the opaque liquid that's often found in infected tissues. Now, in order to phagocytose extracellular material, both macrophages and neutrophils express a repertoire of surface receptors that bind to extracellular debris such as microbes. These receptors include things like dectin-1, which recognizes fungal wall components, as well as mannose receptor, which recognizes glycoproteins that are only found in microbes, as well as Fc and complement receptors, whose function we'll cover more later on. Collectively, these molecules are something that we call phagocytic receptors. Now, once a phagocytic receptor recognizes and binds to its cognate ligand, they trigger a signaling cascade that allows the cell to engulf the extracellular target and transport it inside of the cell, forming a membrane-bound organelle called a phagosome. This process requires actin polymerization and the Rho and RAC GTPases, though we won't discuss this further in this course. Now, once the phagosome is formed, it can fuse with acidic lysosomes that contain various molecules that are toxic to microbes, forming something that is logically called the phagolysosome, where microbes can be broken down and killed. Phagolysosomes are also important sites of microbial recognition through pattern recognition receptors, which we'll learn about in the next lecture for this week. This table from the textbook summarizes the mechanisms used by phagocytes to directly kill pathogens. These include molecules that are present in the phagolysosome that we just discussed. These are acidic organelles with a low pH that can function by being either bacteriostatic, meaning that it halts bacterial growth, or bactericidal, meaning that it kills a bacterium. Phagolysosomes also house toxic oxygen intermediates that are also referred to as reactive oxygen species, or ROS. These include molecules like superoxide and hydrogen peroxide that are highly reactive with other molecules and can help tear apart ingested microbes. Just as a side note, NADPH is a key enzyme used in the generation of ROS. And so phagocytes also exhibit something called the respiratory burst, where they rapidly consume large amounts of oxygen once they're activated in order to generate these reactive oxygen species. In addition to ROS, there are also reactive nitrogen species, or RNS, such as nitric oxide, which can kill microbes through similar mechanisms to ROS. Phagocytes can also kill pathogens through the production of molecules that are secreted either or both into the phagolysosome as well as into the extracellular space. These include some of the antimicrobial peptides that we learned about in our discussion of physical defenses at the epithelial barrier, including cathelicidin as well as alpha and beta defensins. These are amphipathic peptides, which means that these are small proteins that have both hydrophobic and hydrophilic regions, and this allows them to insert themselves into microbial cell walls and membranes in order to form pores that subsequently kill the pathogen. Lastly, there are a number of enzymes secreted by phagocytes that can chew up microbial components, including the lysozyme found in tears, as well as a variety of other proteases that interfere with microbial survival. taking all of these mechanisms into consideration, we can really appreciate the diversity of methods that macrophages and neutrophils can use to kill harmful microorganisms once they've been nibbled up by phagocytes. The last major function of innate immune phagocytes that we'll cover today is their ability to produce inflammatory signaling molecules that generate inflammation in the body. Now, I know that inflammation is a term that's thrown around a lot lately, especially in the popular press with some pseudoscience and alternative medicine fields. So first, let's define inflammation. In terms of clinical symptoms, inflammation is defined by the presence of pain, redness, heat, and swelling. And these are clinical manifestations of signals that are produced by leukocytes called cytokines and chemokines, which function by increasing the flow of both fluids, primarily blood, as well as cells into infected tissues. And I really want to drive home the point that the primary purpose of local inflammation is to allow for the activation and recruitment of effector immune cell types and signaling molecules to the site of infection, where they can then undergo activation and exert various functions to help eliminate the infection. Now, let's go over some quick specific examples of these inflammatory mediators. Just as an example, I'll talk about molecules produced by a macrophage that's been activated, but please keep in mind that both cytokines and chemokines can be produced by most other cell types, both leukocytes as well as other non-immune cells. First, let's talk about cytokines. These are small proteins that can be secreted in soluble forms or remain tethered to a cell, and the soluble forms can function similarly to hormones in that they can travel through the circulation to influence cells that are far away from their site of production. Although cytokines are a distinct class of signaling molecules in comparison to hormones. Now, common types of cytokines include interleukins, which are abbreviated by L, as well as tumor necrosis factor or TNF family molecules. Interferons, abbreviated through IFN, are another type of cytokine that are especially important for antiviral immune responses, and we'll discuss those a bit more later. Now, just as an example, let's look at the activities of tumor necrosis factor alpha, or TNF alpha, and IL-6, which are two common inflammatory cytokines. TNF alpha can bind to TNF receptors on responding cells, including vascular endothelium. These are the endothelial cells that line the vasculature, and then induces an activation state in these endothelial cells. Now, activated vascular endothelium increases the diameter of blood vessels in order to increase blood flow to a local tissue, resulting in the heat and redness symptoms of inflammation. Activated vascular endothelium also leads to increased vessel permeability, which allows for the increased entry of fluid as well as leukocytes and other signaling molecules into the infected tissue. And these consequences are why we observe pain and swelling at inflamed tissue sites. Due to the influx of fluid into peripheral tissues, this also leads to increased lymphatic drainage, as larger volumes of lymph need to be circulated through the lymph nodes. And this has important implications for the drainage of soluble antigens as well as antigen-bearing APCs into lymph nodes for adaptive immune priming, as we discussed last week. Lastly, TNF signaling also increases body temperature through signaling that's relayed through the hypothalamus, and this results in a fever, which further helps with vasodilation. IL-6 is another major inflammatory cytokine which provides additional important functions in stimulating immune responses. Now, IL-6 receptor activation on leukocytes can help trigger their inflammatory activation so they can execute various effector functions, including increased antibody production by B cells, which we'll learn about in a couple of weeks. IL-6 signaling also stimulates a process called granulopoiesis in the bone marrow, where common myeloid progenitor cells are differentiated into granulocyte progenitors and then subsequently into neutrophils and monocytes, which can then travel through the blood to the site of infection to replenish dead or exhausted phagocytes. And like TNF alpha, IL-6 can also promote fever in the body. In addition to cytokines, activated cells can also produce chemokines. These are essentially a subset of cytokines that specifically promote chemotaxis, or the migration of cells from one site to another. Chemokines typically form gradients where they are highest at the site of production and then obviously diffuse to lower concentrations farther away from the site of production, which allows effector cells to migrate from areas of low chemokine density to high chemokine density and essentially find the source of infection. The naming conventions for chemokines looks a little bit confusing. These molecules all start with either CC, CXC, XC, or CX3C, which is a bit of a pain, but this really simply refers to subfamilies of chemokines that are defined according to the position of sequentially conserved cysteine residues. Now, CC means that the cysteines are directly next to each other. CXC means that they are spaced apart with one other amino acid residue in between. XC means that one amino acid residue precedes one of the cysteines, while CX3C means that two cysteines are separated by three other amino acid residues in between. Chemokine naming prefixes only concern their structure and have no bearing on their actual functions. Now, note that all chemokines bind to chemokine receptors expressed by responding cells, which are always G protein coupled receptors that are located at the plasma membrane. Two examples of chemokines are CXCL8 and CXCL9. CXCL8 recruits neutrophils from the blood, which express the cognate receptors CXCR1 and CXCR2, and this results in the movement of neutrophils from the vasculature into infected tissues. CXCL9 is responsible for recruiting T cells from the blood that express the chemokine receptor CXCR3, therefore resulting in bringing a very important subset of adaptive immune cells to the site of infection where they can execute antigen-specific effector functions. These are just a couple of examples of cytokines and chemokines, but I want to stress that there are tons of other inflammatory and non-inflammatory chemokines and cytokines that can be produced by activated cells, and we'll discuss a lot more about some of these as we move through the remaining lectures in this course as they relate to specific immune cell types and their responses to specific disease contexts. Now, we've established that a key purpose of inflammation is to allow for the recruitment of leukocytes to the site of infection, and that cytokines and chemokines are responsible for the signaling mechanisms that control these processes. But how do leukocytes actually transit from the blood into peripheral tissues? As we noted in our discussion of TNF and IL-6, inflammatory cytokines activate the vascular endothelium that's adjacent to infected tissue, which increases the expression of a variety of adhesion molecules on the luminal side of a blood vessel. These adhesion molecules include selectins, which loosely bind molecules expressed by leukocytes, which makes them kind of loosely tethered to the endothelium, like the monocyte in yellow on this diagram on the right. Once tethered, leukocytes exhibit a rolling behavior, which allows other adhesion molecules to interact, and these include integrins that are expressed by leukocytes. Integrins are named according to different alpha and beta subunits, as well as with some easier abbreviations. So some common integrins include LFA1, this is integrin alpha L beta 2, as well as CD11B, which is integrin alpha M beta 2. Integrins form a much tighter bind with their cognate ligands compared to selectins, which stops the leukocyte from rolling along the luminal side of the blood vessel any further. And once stopped, the leukocyte can then squeeze in between endothelial cells in a process called extravasation, which we also refer to as diapedesis. And in this example, once a monocyte successfully enters the tissue, it then differentiates into a macrophage due to the presence of macrophage differentiation signals. And then it can carry out essential functions of phagocytes that we went over earlier in today's lecture. Now, I want to end today's talk by stressing that inflammation is a critical process in promoting immune responses, but that it's extremely important to limit inflammation specifically to the local site of infection. And I think that this point is best exemplified by learning about sepsis, which is a pathology that results from what's basically uncontrolled inflammation that's present everywhere in the body. Now, sepsis occurs when there is a systemic infection, such as bacteria that are present in the blood that are then allowed to circulate throughout the entire body. This usually occurs when a local infection is unsuccessfully controlled and bacterial spread is allowed. And some common pathogens that are implicated in a lot of sepsis cases are Staph aureus, Strep pyogenes, and E. coli. Now, on the left columns of this figure, we have a summary of inflammatory processes that occur successfully during local infection, where only the site of infection experiences fluid egress and leukocyte extravasation, followed by a wound healing process where the blood vessels are then closed off or occluded. However, during sepsis, because inflammatory signals are being produced everywhere in the body instead of just one location, fluid is removed from the blood everywhere in a process called systemic edema. And this sudden drop in blood volume then leads to vessel collapse and occlusion everywhere in the body, which has a lot of really dangerous effects, including just widespread organ failure. Now, sepsis is an extremely dangerous condition with very high mortality rates outlined here. And I want to note that in experimental models of septic shock that we use to study this form of uncontrolled inflammation in preclinical animal models, such as mice, these involve administering drugs that stimulate systemic overproduction of the inflammatory cytokines that we talked about today, including interferons, IL-6, and TNF. And again, this brings up a central theme that we've harped on a couple times by now, that the proper resolution of inflammation and the return to homeostasis following infection is a really essential phase of an effective immune response. And here it's demonstrated by the fact that the production of inflammatory signals such as cytokines needs to be very tightly controlled. So in summary for today, I hope that we've established the key point that the innate immune system is required to provide immediate defense from infection by any of a number of species belonging to these four major categories of pathogens. We've learned that innate immunity does this through both physical epithelial barriers that provide mechanical, chemical, and microbiological components of defense, as well as through the activation of innate immune sentinels such as phagocytes, which function by phagocytosing and killing pathogens and promoting inflammation. We've defined inflammation as a set of processes that all serve to help recruit and activate leukocytes and other immune molecules to the site of infection, where they can then begin the next phases of an immune response. So that's all for today's lecture, but please remember to watch the remaining 2 lectures for week 2, which will introduce us to other components of the innate immune system.